All right, it's my pleasure to introduce our third speaker for today, this is Dr. Sarah Turner. She spent 10 years working on a wide variety of grant-funded and industry contract research projects in the University of Maine Aquatic Animal Health Lab and went back to graduate school just before the COVID-19 pandemic. She graduated with her PhD in aquaculture and aquatic resources from the University of Maine in May of 2024, where her interdisciplinary research focused on examining cellulose nanomaterials, a biopolymer primarily produced from wood, as a novel adjuvant and delivery system in Atlantic salmon. Very interesting topic, and we're looking forward to the presentation.
Thank you, Nick. Hopefully my slides go a little smoother. It looks like we're having some Wi-Fi troubles today here in Maine. But welcome. Today I want to give you a broad overview of my doctorate work on cellulose nanomaterials as a novel adjuvant and delivery system for aquaculture vaccine applications. This is an interdisciplinary project formed through a collaboration between the University of Maine Cooperative Extension, the Aquaculture Research Institute, and UMaine College of Engineering, and it is funded by a USDA AFRI foundational grant. So according to FAO, aquaculture is the fastest growing food sector, with farm fish production estimated to have reached approximately 296 billion US dollars this year, as an all-time record high, as wild-caught fisheries remain static since the late 1980s due to overfishing, as most of us know. And because of this, aquaculture has been responsible for continued growth in feeding increasing human population. Within the aquaculture industry, Atlantic salmon are within the top 10 species farmed globally as a high-valued protein source rich in omega-3 fatty acids. However, infectious disease in fish represent a major economic issue in aquaculture worldwide, and this accounts for approximately 40% loss in production potential, estimated at $102 billion in losses. These pathogens represent a significant hurdle for current and future growth of the aquaculture industry, such as Vibrio angularum, a bacterial pathogen of salmon, causes these large lesions seen in this image, and was the target pathogen of this research. Because of the negative impact of these pathogens, the aquaculture industry is dependent on vaccines to remain viable. And again, unfortunately, current oil-based adjuvanted vaccines can vary in duration of protectiveness. They can be expensive and come with negative consequences in the fish, including growth penalty, potential residue issues, autoimmunity, and significant adhesion formation and pigmentation around the injection site. Again, Atlantic salmon were our target species because they are a high-valued farm food source that are also susceptible to a host of diseases. They're vaccinated regularly and as such impacted by the adverse effects of vaccination. But the overall takeaway message here is there is a growing need for a new generation of vaccine technology in the aquaculture industry to sustain future growth. So there are essentially two groups of nanosystems that have been previously examined in finfish adjuvanted vaccines to improve upon vaccine efficacy. Inorganic metal oxide nanoparticles, such as zinc oxide, are generally non-biodegradable and toxic. And within organic nanosystems are a class of materials called biopolymers. Biopolymers such as chitosan, PLGA, alginates, and others are readily available polysaccharides. And these biopolymers at the nanoscale generally avoid environmental and toxicity concerns and have shown great initial success as vaccine adjuvants in a variety of finfish and terrestrial mammals. Conveniently, we produce the most abundant biopolymer on Earth at the nanoscale right here at the University of Maine, nanocellulose, which is also called CNM, cellulose nanomaterials. This material is sustainable, biodegradable, biocompatible, and highly tunable cellulose at the nanoscale. And it can be manufactured in industrial quantities at relatively low cost. It's very stable for production and storage, and overall meets the rigid medical requirements of a suitable vaccine component. CNM can be sourced from anything containing cellulose, with the most predominant source being wood fiber, but also from other plants, marine tunicates, and bacteria. One modification that can be performed during manufacturing is to replace the surface hydroxyl group with a carboxyl group, and hopefully you can see that here in red. And this produces a form of CNM called tempo-oxidized cellulose nanofibers, which I'll just refer to as TEMPO. The benefits of TEMPO are that it's produced in large quantities using green chemistry, to remain in aqueous suspensions, and it can be modified in a lot of ways that other forms cannot because of the presence of that surface carboxyl group. So in other words, that carboxyl group allows for high tunability with high surface area and negative charges, making antigen carrying and loading with high stability feasible. These properties can make CNM an attractive material for novel aquaculture vaccine systems that have not been examined in finfish prior. So we collaborated with the University of Maine Biomedical Engineering Department, as UMaine Engineering is well suited for nanotechnology research to produce stable hydrogels from Tempo, as they have the Furland Engineering and Education Design Center, brand new facilities and expertise in this area. So we essentially tuned CNM in 3 different ways. The first was unmodified tempo, and the second was we used physical cross-linking of the tempo to form a stiff hydrogel. And this was delivered into the peritoneal cavity, similar to a pit tag, using a pit tag implanter, with the idea being that it could release antigen over a long duration of time in a controlled manner. The third form was to chemically modify tempo, so it's injectable through a small 26 gauge needle, and then cross-linked after injection into a stable hydrogel in the peritoneal cavity, and again to control release antigen over time. So after production and characterization of the vaccine formulations in vitro through our biomedical engineering team, I performed vaccine development research in Atlantic salmon in our high containment wet lab facility. Again, this facility allows for industrial collaboration and product development, specifically for advanced vaccine research in areas of safety, antibody response, and efficacy studies in finfish. So first, safety studies were completed in random block duplicate rack systems in Atlantic salmon par to assess the risk of harm to health as a result of adverse reactions from CNM. Various formulations of CNM were compared to a commercial oil-based adjuvant positive control, as well as a negative PBS control. Fish were acclimated, baseline sampling was performed, and then they were vaccinated with the formulations. Fish were necropsied at time points to collect biometric data and examined for adverse tissue reactions. Safety metrics were examined, including condition factor to monitor for changes in growth, and then adverse pathology scored by eye using the Spielberg scoring rubric, as this is a well-accepted rubric for vaccine studies to assess formation of adhesions, melanization, and residue. And then finally, tissues were collected for histology. Results showed repeated safety of unmodified tempo with the ability to remain in the peritoneum for the entire duration of the study with minimal adverse reactions. Additionally, fish maintained vigorous feeding and normal behavior during the studies with no significant differences between treatment after 600-degree days. Internal adverse reactions and histopathology scores were determined to be dependent on how much the tempo CNF was cross-linked, such that reactions range from none or normal pathology in low cross-linking tempo to the development of severe granulomas and inflammation surrounding the CNM hydrogel, as you can see here, when it was highly cross-linked physically into the implantable hydrogel. Unfortunately, for chemically modified injectable hydrogels, Kaplan-Meier time to death curve showed significant acute mortalities in our 2x chemically modified formulations seen in the pink and the orange. And the unvaccinated fish, the negative sham controls, and unmodified tempo had a reduced risk, reduced risk of mortality compared to oil adjuvanted positive control. And I want to note that the 1x chemically modified tempo shown in the green did have greater mortality. However, this was not significantly different from the positive control. In addition to mortalities, we also saw gross and microscopic pathologies consistent with acute toxicity. However, for time, I'm not going to show those results, but obviously this is unacceptable for the industry. So next, we quantified antibody response to vaccination. As it's been shown, the main systemic antibody in finfish, IgM, can increase 2 to 4 times in response to oil-based vaccination. So this was performed in 3 replicate cohabitational tanks where fish were acclimated for 14 days. baseline sampling was performed, and they were vaccinated with either one of 5 different CNM vaccines or an oil-based commercial vaccine as the positive control, and again, PBS for the negative control, and then sampled over time to look at total IgM gene expression. And also serum IgM levels were collected for antigen-specific indirect ELISAs. So RT-qPCR results performed for gene expression from the head kidney and the vaccinated fish. Overall, total IgM expression was upregulated in unmodified tempo, as well as some other modified tempo formulations, alongside the positive oil adjuvanted control at both time points, compared to the negative control by Dunnett's post hoc analysis. And this is very promising, as again, IgM is the systemic antibody that increases in response to vaccination. I'd also like to note that I did look at IgT and IgD expression from the head kidney with no significant differences in upregulation or downregulation between formulations. Preliminary data using indirect ELISAs quantified the serum IgM titers specific to antigen in the matched gene expression samples and suggested an increased IgM response specific to the Vibrio angular antigen. This is promising. However, we had a fairly small sample size and a 9 with some large variation. So we're currently processing more samples for analysis. So where did we go from here? Well, so what we did was our chemical modification of tempo. We actually found some ingredients, specifically ionic salts that are readily available and safe. And those salts showed, when cross-linked with the tempo, showed safety after 600 degrees post-injection in the Atlantic salmon. And as you can see, it looks like a clear liquid and injects like a clear liquid, but after injection, it cross-links into a stable hydrogel, as you can see stained in the Kamasi blue in the lower picture. And again, our next steps would be to perform a challenge efficacy study to demonstrate vaccine protection under disease conditions, which I hope to perform this fall. And if successful, this vaccine technology could translate to other farm fish species or terrestrial animals. So overall, in summary, unmodified CNM, specifically tempo, demonstrated repeatable safety evidenced by low risk of harm to health. The 3D physically cross-linked tempo hydrogels were difficult to deliver, and this resulted in a strong innate immune response, making it an unlikely candidate for vaccine adjuvants. Our chemical modification to tempo was injected through a small gauge needle to form a matrix in vivo, but trace components remained. And CNM is a promising new adjuvant for aquaculture that may induce an IgM antibody response, while using ionic salts could produce a similar mechanism for a safe and shear thinning vaccine efficacy study needed. And with that, I would just like to acknowledge Dr. Inga Seidor for our histopathology, as well as Dr. Michael Habitzian and Dr. Zhanghui Hong, and all of our staff and students here at UMaine. And we'll take any questions now. Thank you.
Great. Thanks a lot, Sarah. Very interesting approach. Folks, if you have a question, feel free to unmute yourself.
Sarah,
Yeah.
I have a simple question. This is Gail Curath out in Seattle. I might be, it's just that I have not worked in vaccines for quite some time. The unmodified tempo, is it that cannot be used? Is there any possibility that that might work, I guess is my question.
Yes, it is a possibility that unmodified tempo could work. However, we did do some preliminary antibody response studies, which I didn't show the data for. And I think what happens is because the unmodified tempo has a negative charge and our bacterial antigen also has a negative charge, we tend not to see consistent IgM antibody responses with it. And so with the cross-linking, it just helps to hold that antigen there And as the tempo starts to uncross-link or slowly degrade, it releases the antigen to improve the antibody response a little better. So short answer is unmodified tempo is a possibility, but I think with the cross-linking, we could improve consistency.
Great, thank you.
Yep.
Awesome. Thanks again, Sarah. Virtual
Yep.
round of applause to you.